And here we are back with the digestive system, but focusing in on the small intestines. So the small intestines, here we're going to have almost all of the chemical digestion and nutrient absorption that's going to occur. It is called the small intestines because of the diameter, not the length, because it's actually very, very long. It's actually the longest part of the digestive tract, about 5 meters long in a living person. And it begins at the pyloric sphincter at the very end of the stomach, all the way to the ileocecal valve that's going to open to the large intestines. And so here you can see the sections of the small intestines. The duodenum is going to begin right at the beginning, and it's going to kind of be tucked away behind those large intestines there. And then that purple portion here is the jejunum, and then this more pinkish area is representing the ileum, and then you have the meeting of the large intestines here. So here, those are in a cadaver. You can see the large intestines going all the way around that abdominal cavity, and then the small intestines highlighted in that teal color. And what's connecting and holding on to those small intestines is the mesentery. So the small intestines, it begins at the pyloric sphincter. And then it will arc around the head of the pancreas and it passes to the left. It's going to end at a sharp bend that is called the duodenal jejunal flexure. Right, that's a mouthful. And so slightly distal to the pyloric valve, it exhibits that previously described wrinkles called the major and the minor duodenal papillae, where it receives the pancreatic duct and accessory pancreatic duct, respectively. So here is that picture again, and you can see here's my laser pointer. You can see the gallbladder here, and then you can see those hepatic ducts that are coming from the liver. They will converge to form the bile duct, and then that will empty its bile or contents into the duodenum, into this section here by that major duodenal papillae. Remember, papillae, the root means nipple, so those are these nipple-like projections that stick into the duodenum. And then you can also see there is the pancreatic duct that goes through that main portion of the pancreas, as well as the accessory pancreatic duct. So the pancreatic duct that will end up emptying sodium bicarbonate substance into that duodenum through the major duodenal papillae, but the accessory duct that's going to go over here and empty its contents into the minor duodenal papillae. And then you can see that this section of the duodenum is fairly short and it wraps around the head of that pancreas, and then it does a sharp turn at that duodenojejunal flexure, and then it's going to become the jejunum. So the duodenum, most of the duodenum, along with the pancreas, is retroperitoneal. So what that means is that it is situated or behind the peritoneum. The duodenum receives the stomach contents, pancreatic juices, and bile, and the stomach acid of the chyme will be neutralized here by the sodium bicarbonate that's going to be released from the pancreas. Bile that breaks down fats, so bile that's secreted from the liver or stored by the gallbladder will be released and eventually end up here, and it will physically break up fat. That process of physically breaking up fats is called emulsification. So these fats will be emulsified by the bile acids. And then we're going to have pepsin that will be inactivated, because remember, pepsin was activated and converted from pepsinogen by the presence of hydrochloric acid and that lower pH. So now we have sodium bicarbonate that's a high pH, and that will then inactivate this pepsin because of the elevated pH. And then we'll have pancreatic enzymes begin to take over the job of chemical digestion. And in this image, you can see the peritoneum, and you can see the pancreas here. And then you can also see the duodenum right here, and showing that it is actually behind this peritoneum. So once we make that turn past the duodenal flexure, we're now going to be in the jejunum. The jejunum is going to make up about 40% of the small intestines after the duodenum, and it's going to be about 1 to 1.7 meters in a living person. And so if you were to take a cross section of the small intestines, like cut the jejunum right through the tube, so that way you can look down the lumen or through the tube, what you would notice is that it doesn't look smooth. It actually kind of looks like cardboard looking. And it has really thick muscular walls. So those projections that stick up into the lumen to give it that rough appearance is these large circular folds. And what those circular folds are doing is increasing surface area, because rather than being smooth, they're going up and down and providing these projections, increasing the surface area that food can come into contact with. The walls are also going to be really, really thick and muscular, which makes sense because we're going to have to segment the food and physically break down the food and mix it. We're also going to have to push that food through the tube. So we're going to need some thick muscles for that. And then it's really reddish in appearance, and that's because it has a rich blood supply, which should make sense because we're doing a lot of nutrient absorption here. So in order to absorb those nutrients and get them delivered to all the cells in the body that need those nutrients, we're going to have a lot of blood ready to pick up those nutrients and deliver them. So we're going to have most nutrition absorption as well as digestion that will occur here. The last 60%, oh, I just realized I have a typo right here. It's supposed to be small with 2 L's. But the last 60% of the small intestines is the ileum. And it's about 1.6 to 2.7 meters in length. And what we're going to notice is when we're in this section of the small intestines, the muscle layer is actually a little thinner. It's not as thick, or it's less muscular. So we're going to see that those tubes are actually thinner. And it's going to be a little paler in pink because it's not as vascular, since we're not absorbing as many nutrients here. And as mentioned in the lymphatic system, we're also going to start to find what's called these Peyer's patches. These are those prominent lymphatic nodules that we will find occurring in clusters that are on the opposite of where the mesentery attaches to the intestine. And then at the very end, we're going to find a valve called the ileocecal valve. This is where the ileum is going to meet with a section of the large intestines, this pouch-like structure called the cecum. So we will have the ileocecal valve. It is not really considered a sphincter because it's not round, so we're calling it the ileocecal valve, even though some resources and some literature will refer to this as the ileocecal sphincter, but it's formed from these thickened muscularis layers in the ileum, and it will regulate passage of food into the large intestines. And then when that large intestines, that cecum portion gets filled, that will actually pinch this valve shut. And this portion is intraperitoneal along with the jejunum, which means that it is just within that peritoneum, unlike being retroperitoneal like the duodenum. And because it is intraperitoneal, it will be covered with a serosa. So here is a nice diagram that shows you the diameters of both the small intestines here and the large intestines. So you can see that large intestine diameter is definitely much, much larger. And then you can see where they meet, there is that ileocecal valve. And you can see that food could pass through and go in, filling in this area right here. That would be the pouch-like structure called the cecum. And as this begins to fill up, it will pinch this ileocecal valve shut, preventing food from going from the large intestines backwards into the small intestines. And then you can also see here, there is the veriform appendix. That's your appendix, which has some thought to previously have some lymphatic function, but it doesn't really serve much purpose for us now. Here we have a good diagram that just shows you an overall picture of the entire digestive system, and it shows you those large intestines along with the small intestines. So when looking at the microscopic anatomy of the small intestines, we're going to see the same layers that we've seen previously. We're going to see the muscularis layer, the serosa, mucosa, the submucosa, okay, all of those. So the innermost layer of mucosa is responsible for nutrient absorption. So if we think about what we learned in 201 with tissues, the type of epithelial tissue that is specialized for absorption is a simple columnar epithelium. So that's what we're going to find in this innermost mucosa layer. We're going to find these one layer of these tall column-shaped cells. And what we end up finding in between the simple columnar epithelial cells are specialized cells called goblet cells that can secrete mucus. And in some areas, we're going to find the cells are topped with these little hair-like projections called cilia that can help move that mucus through the tract. The submucosa layer is going to contain blood vessels and glands. And the muscularis layer is going to use smooth muscle for movements. So if we look at the muscularis externa, it is noted for having a thicker inner circular layer and a thinner outer longitudinal layer. So the circular layer, when those muscles contract, they will squeeze, decreasing the diameter of the tube. While the longitudinal layers, when those muscles contract, they will shorten the length of the tube to push things through. And then we have the outer serosa layer that's going to provide a protective kind of outer coating. So we have a large internal surface because we have many folds that are increasing the surface area. And the circular folds that we talked about before are the largest of these elaborations. They're these transverse spiral ridges that are actually up to 1 millimeter in height. And they involve the mucosa and the submucosa. So they will not be visible on the external surface, which is smooth. These guys, because they stick out so much, are going to slow the progress of the chyme and make it flow in somewhat of a spiral pathway. And that's going to increase its contact with the mucosa and promote a more thorough mixing and nutrient absorption by that contact. The circular folds are going to begin in the duodenum, but in the jejunum, they're especially large, tall, and they're going to be very closely spaced. And then they're going to become smaller and more sparse in the ileum. The changes that we see in these circular folds are correlated with the relative amount of nutrient absorption that is happening in each region. Circular folds are going to be absent from the distal half of the ileum because most nutrient absorption is completed by that point. So that kind of makes sense. And then we're going to have these villi, and villi projections, they are what give the inner lining of the intestines kind of a fuzzy-like texture. They're kind of making it look kind of like a terry cloth, like one of those fuzzy cloths. They're about 0.5 to 1 millimeter high, and they have these tongue to finger-like shapes. The villi are going to be largest in the duodenum, and then they will become progressively smaller in the more distal regions of the intestines. And the villi will be covered of one of our two types of epithelial cells. They'll either be those mucus secreting goblet cells, or they'll be those columnar epithelial cells. We call those enterocytes, and they are the cells that are actually doing the absorption. So they are the absorptive cells. So they're just like the epithelial cells of the stomach. Those that are in the small intestines are going to be joined together by tight junctions that prevent digestive enzymes from seeping in between them and eroding all of the underlying connective tissue. Then we're going to have our microvilli, which are going to be much smaller extensions, and they're actually extensions of the plasma membrane, and they're only about 1 micrometer high. So they're really, really, really tiny, and we'll talk more about those in a bit. Here you have a decent diagram that shows the small intestines and some of the layers. But what I want you to look at are these circular folds here. And then you can see these finger-like projections of the that are making those circular folds. And then the smaller projections here, these little finger-like ones, those are the villi. What we're seeing in image A here, this is actually a scanning electron microscope image of each villus. It is about 1 millimeter in height. And then in B, you can see the layers in the histology. So you can see that muscularis layer, and then we're going to have the longitudinal and the circular muscles found in there. And then we're going to have that submucosa layer, and then we'll have the mucosa layer. You can see those villi, and then deep in between the villi, you have a structure called the intestinal crypts. If we look at structure C, we are looking at the structure of villi, and you can see that there is that really rich blood supply. So you're going to have arteries and veins, as well as lymphatic vessels that are running through here. And then you can also see those goblet cells that are in gray. You have the absorbing epithelial columnar cells, those enterocytes that are in pink there. So if we keep looking at the microscopic anatomy and we zoom into one of those villi, well, the core of the villus is filled with an areolar connective tissue of the lamina propria. And it contains an arteriole, those blood capillaries that we saw, a venule, and a lymphatic capillary that's called a lacteal. The blood capillaries are going to absorb most of those nutrients, but the lacteal, that is going to absorb most lipids. And lipids will give its contents a milky appearance, for which the lacteal gets its name. The core of the villus also has a few smooth muscle cells that will contract periodically, and that will enhance the mixing of the chyme in the intestinal lumen. And it kind of does a milking motion that milks the lymph down the lacteal to larger lymphatics in the submucosa. So here is that picture. Again, if we were to zoom into one of those villi, we can see those goblet cells, the absorbing enterocytes, and then here you have that lymphatic lacteal that's going to absorb the lipids and carry it down to join the lymphatic vessel. The microvilli are going to form a fuzzy brush border on the surface of each one of our enterocytes, those absorbing cells. So by having this brush border, they're like little hairs on top of those finger-like projections. So that's additionally increasing our surface area. But they're also going to actually contain an enzyme that's called a brush border enzyme that's part of their plasma membrane. So they don't actually secrete this enzyme into the lumen of the intestines, but these enzymes are going to be part of the cell membrane. And they carry out some of the final stages of chemical digestion. So the chyme must come into contact with the brush border for the digestion to occur. This is a process that is called contact digestion. And this is one of the reasons it's so important that the intestinal contractions churn that chyme and ensure that it all contacts the mucosa. Otherwise, it will not be completely digested. So here we have some electron images of the intestinal microvilli. So if we just zoom into this one structure here, this is one of those enterocytes, the absorbing cells, and you can see those little hair-like projections that are called the microvilli. And then on the floor of the small intestines, between the bases of our villi, there are going to be numerous little openings or pores, pores that go into the tubular glands, and these are called intestinal crypts. These crypts are similar to gastric crypts or the gastric glands, so they will extend as far as the muscularis mucosa. And on the upper half of this duct or crypt that's going into the mucosa, the upper half is going to consist of those enterocytes and goblet cells, like those of the villi. But the lower half is going to be dominated by a different type of cell. It will be dominated by dividing stem cells. And these cells only have a lifespan of about 3 to 6 days. And as the cells in the underlying layer divide, they're going to push the cells on top of them up. And these cells migrating up through the crypt to the top of the villi will eventually be slushed off and broken down during digestion. At the base of the crypt, we're also going to find these other types of cells called pineth cells that secrete lysozyme, phospholipase, and defensins, which are just defensive proteins that resist bacterial invasion of the mucosa. And ignore my typo right there for Peyer's patches. So here, if we look at the duodenum and we can see the different layers, you have the mucosa layer, which is this layer right here. And then if we want to focus in on just a villi, it would be one of these guys. And then those intestinal glands will be found right down in here. And then we've got our submucosal layer, that is this layer here. And we're going to find Brunner's glands down in the deeper portion of that submucosa. And we're also going to find ducts of the submucosal glands kind of pushing up into the mucosa layer there. And then we have this lowermost layer that is going to be the muscularis externa. And you can see that really, really thick circular layer and then the thinner longitudinal layer there. If we look at the ileum, we can also see some of those layers. You can see the mucosa layer, and it's a pretty thick mucosa layer, and we can zoom in on just one of these villi. The submucosa layer is going to be this layer here that is deep to that mucosa. And then we can also find some lacteals. That would actually be like this portion right here, in this area here, pushing up into the middle of those villi. And they will again absorb those lipids. And then we have those Peyer patches that are going to be found deep in the mucosa layer here. So right here, you can see this kind of little patchy kind of purple area. And here we have the ileum. So the ileum, you can see that there is one layer of those column-like cells. So that is that simple columnar epithelium, and this is an enterocyte. And then you can see a goblet cell, which is actually the cell right here, and this is the droplet of mucus that he's secreting. It's actually really kind of a good picture. You can see the mucus spewing out of that goblet cell. And then you can see the kind of fuzziness right along the lumen. This open space is the lumen. So this kind of fuzziness, that is the brush border there. And here's another really nice picture of one of those goblet cells. So the cell is actually right down here. And this is the mucus that he is secreting to be released. And then you can see that nice brush border right there as well. For the ileum, we're also going to have those Peyer's patches that we can find in the submucosa of the ileum. And this is actually a really good picture because you can see those patches really nicely surrounded by the darker stain. So as the intestines move food, they're going to move food through them by peristaltic contractions. So you're going to have the circular folds decrease the diameter of the intestine, and you're going to also have the longitudinal muscle decrease the length, and that's going to propel food through the intestines. So when we have contractions of the circular muscle and contraction of the longitudinal muscle, that's actually going to give us more than one function. It's going to give us three. One is going to be to mix the chyme with intestinal juices, with the bile and pancreatic juices. And by mixing chyme, we're going to neutralize that stomach acid and break down nutrients more effectively. Because whatever the acid didn't break down, the basic nature of the sodium bicarbonate will, or if it is fat-based, that bile is going to get it broken down. And as we churn that chyme, we're going to bring it into contact with that mucosa layer for the contact digestion that we were talking about. We need the chyme to contact the brush border because those enzymes are not secreted out. So for final breakdown of the nutrients, we need them to come into contact with the mucosa layer. Also, by coming into contact, that is how the nutrients get absorbed. If there's no contact, the nutrients can never diffuse through the epithelial tissue and into the blood supply that we find in the villi. So we have to have contact for those nutrients to get into the body. And then we're also going to have movement of the residue towards the large intestine. So when we talk about motility, there is segmentation and peristalsis, and they're very often confused. Segmentation, this is going to be mixing. So we have movement, which will not move food forward through the tube. It's going to kind of stay in the same place. So we're going to see these stationary ring-like contractions that appear in several places along the intestines. And this is the most common type of intestinal contraction. They occur about 12 times a minute in the duodenum, and then it slows down as we get to the ileum, about 8 to 9 minutes. But what's controlling the pace of this contraction are pacemaker cells that are inside of the muscularis externa. And when most of the nutrients have been absorbed through that, segmentation is going to start to decline, and we're going to start to see peristalsis begin. So this image is showing progressive stages from the top to bottom. You have the circular contractions of the intestines that are going to cut into the contents. And by cutting into the contents, it churns and mixes them as represented by the progressive blending of the two colors. And once most of these nutrients have been absorbed and little remains, but really undigested residue, that segmentation will decline and peristalsis will start to begin. So the duodenum will secrete a hormone called motilin, and motilin triggers a peristaltic wave beginning in the duodenum. And that wave will travel about 10 to 70 centimeters, and then it will die out, only to be followed by another wave that begins a little bit further down the track than the first one. So these successive overlapping waves of contraction will pull and kind of milk the chyme towards the colon over a period of about 2 hours. And so these waves, these overlapping waves, are called a migrating motor complex. So the migrating motor complex of peristalsis is where we have these successive waves that are overlapping each other. And each wave will travel partway down the intestines and drag or milk the contents towards the colon. And once they meet the small intestines meets the large intestines, we will come to a valve called the ileocecal valve that is usually closed. Food, though, when it gets into the stomach, it will trigger the gastroileal reflex. And that gastroileal reflex will enhance segmentation in the ileum and relax the valve. So that way the food will start to get pushed and fill the cecum, which is that first part of the large intestines, that kind of pouch. And then as the cecum fills up with residue, it's going to increase the pressure within the cecum, and that will pinch the valve shut. And by pinching the valve shut, we prevent reflux or the movement of the contents back into the ileum. We don't want things moving backwards, only forward. And so we will end it here, and I will talk to you more next time.